Hello everyone, today we are going to present you the characterization of clones. But first, what is cloning? It is basically we produce a recombinant DNA and transform into a host organism, then we multiply into thousands and millions of copies. Diagram below shows the process of cloning, and there are 6 steps of cloning. Alright, step 1. Choice of host organism and cloning vector. To choose a host, specialized application may call for specialized host vector systems. So there are 3 types of hosts. First is bacteria such as E. coli, second is yeast, and third one is mammalian. So to choose a vector, it must contain vector itself must contain markers and restriction enzyme site. So there are 4 types of vector: plasmid DNA, bacteriophage, cosmic, and lastly, artificial chromosomes. So step 2, the preparation of vector DNA. To extract DNA, we need to lyse the cell, then remove the lipid. After that, deproteinize the cell extract, and lastly, remove the RNA as well. So the diagram below shows the process of extraction, and on the other side there are 6 different types of methods to isolate the plasmid DNA. So take a look each and every types of plasmid DNA isolation method. Step 3. The preparation of DNA to be clones, which is mainly talking about PCR. So PCR is a polymerase chain reaction. So actually we need 5 components in PCR. Alright, let's enjoy the video as it tells us about PCR in details.
Polymerase chain reaction, or PCR, uses repeated cycles of heating and cooling to make many copies of a specific region of DNA. First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands. When the temperature is decreased, short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence. The primers bracket the target sequence to be copied. At a slightly higher temperature, the enzyme Taq polymerase, shown here in blue, binds to the primed sequences and adds nucleotides to extend the second strand. This completes the first cycle. In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies. After 3 cycles, the target sequence defined by the primers begins to accumulate. After 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.
Besides that, there are another 6 types of special PCR technique. So take a look each and every types of them. Step 4. Creation of recombinant DNA with DNA ligase. So basically this step is use ligase to join all the DNA fragments together. Step 5. Introduction of recombinant DNA into host organism. Usually there are 3 types of gene transfer in prokaryotes. Transformation, which is transfer the genetic materials from dead bacterium bind to the recipient's bacterium. Transduction, bacteriophage transfer genetic materials from one to another. Conjugation, direct physical interaction between 2 bacteria. On the other side, eukaryotes has categorized into biological and non-biological. So take a look at that. Besides that, there are 4 special gene transfer techniques. Slides after this are the advantages and disadvantages of different types of techniques. So before we show you the last step, let's enjoy this video about micro-injection. Step 6. The screening of clones with desired DNA inserts and the biological properties. Actually, this step is all about DNA analysis and also to test the resulting clones for the presence of the insert in the vector. Firstly, blue-white screening is a rapid and convenient detection of recombinant bacteria. Second, gel electrophoresis is used to separate RNA and DNA molecules based on their molecular weight.
Thirdly, softened blot is used to detect the presence of DNA in sample.
And lastly, DNA sequencing is used to determine the precise sequence of nucleotide base in DNA fragment. So the most common type is the Sanger method. Alright, this is all from us. Thank you for watching.